So now let's talk about gene ontology. So gene ontology or enrichment is the functional annotation of your gene, what kind of functions they perform. So basically as we say, we have three kinds of gene ontological function. You have, for example, if we see types of gene ontology, if we search for that, we will have molecular function, that describes the molecular activity. We have the biological process that describe the cellular or physiological role of the genes. Then we have the cellular component describing the location in cell where the gene product produce and execute its function. So we can find three types of function. So now let's just focus on data preparation. To begin the analysis, we need to identify unique gene identifier, which are also ensemble IDs. So where we can find them? We can find them in the gene page. So we will move on to the, sorry about that, we will close this and open the NCBI gene database. After opening the NCBI gene database, we will search for our genes. So for example, I have a list table here, and then I'm just gonna search for the RAPA-P1A gene. So after searching for this gene, we have to find the unique identifier. So here is the ensemble ID or the unique identifier, you can see it. So I'm just gonna copy this unique identifier and then paste it here in our Word file. Unique identifiers. You can also call them ensemble IDs if you want. So here is the ensemble ID or unique identifier of that. So this is how you can discover the identifiers. So I already have list of identified organisms that we are going to deal with. Not organism, but the list of genes, differentially expressed genes that we have. So here we got the 5 genes, and here is the 5 unique identifiers for those genes. You can look up at the database for those if you want to. So now let's just see how to perform the gene ontology function. So if we move here, we will see to elucidate the biological process, cellular component and molecular function influenced by a lung cancer, a gene ontology enrichment analysis was performed. The Shiny Go online tool was used for this analysis, and the gene identifier was used as input. After that, we have the maximum capacity of 20 pathways and 2 pathways were considered. So let's just say we have 2 types of tools. I'm just gonna introduce you to those. So first we have Shiny Go. You can search that, and it is developed by South Dakota State University's professor. It is a very good tool, so you just have to paste your ensemble IDs and submit, and your analysis will be done and shown here. So, but there is a efficient way to do this by using the R programming language. So we will go to the gene ontology database, which is the gene ontology resource Go database. So here we will see Go Enrichment Analysis tab, your gene IDs here. We will paste our gene IDs here, and after pasting our gene IDs, we will search for example, we will search for biological process. So here is a biological process that we searched for, and here you can see we have the analysis type of Panther over representation test gene ontology database, Analyze test, we have the upload file, and the reference list, we have the Homo sapiens, all genes in the database. Annotation test, we have the Go biological process, and the test type, we have applied the Fisher exact test, and correction is false discovery rate. After getting the results, we will click to, we will see we have the unique IDs of 5 out of 5, unmapped IDs none, multiple mapping information none. So we will just export the table. So after clicking on the table, we have the functions table here. So if we open this, here is the results of our information. Here is the results that we need. So how to visualize this? For that, I have developed a code in R that you can use to visualize the results. So for example, here we will move on to gene ontology tab, and if we see, we have to work with a library which is the ggplot2. So ggplot2 is used for the visualization. Then we will have to create a data frame within the results. So here I will create the data frame tab, and the Go term that was identified are represented here. So once you are done with your analysis, you can paste all the Go ontologies like this. So if we open this tab in Excel, for example, so here you can see you have the Go ontologies. Here you can copy all these and then paste it in the code here. From here to here you will get your result and the full enrichment below. After that, we will create the bar plot. So let's run this code. Control plus enter. We will load the library. After loading the library, we will load the data frame, and after loading the data frame, we will also enter the fold enrichment. After that, we will create a bar plot which will show us the gene ontology term enrichment analysis. So if we will zoom on to figure, we have the regulation of T cell migration, GTPase activity. So we have learned about these activities in the previous videos as well. If you see, we were at the CDK gene where we were seeing it is regulated by GPTase. So here you can see the functions of those genes are represented here. So there is also GTPase activity, ERK1, which is similar to the CDNK2 gene, which is our downregulatory gene. So functions are showing here. So once we have this, we will copy this image and move to our results tab and paste it here. Sorry for that. If we can, yes, we will paste them here. So we can do this by using this tool, and it is better if we just save this image in our desktop repository that we created, lung cancer project, and here we will save the file with the biological process .png. So we will move to our repository, lung cancer project, and then we will see if we have the result. So Go biological process are represented here. So we will import these to our result slides. You can slide these here. We have the Go biological process, and you can just say that we have performed this analysis by using the R programming language and using the gene ontology database. So in the B tab, in the B results of the figure, you can include the molecular functions of the genes as well. So we will launch the analysis, After launching the analysis, we will see there are no significant results. But if we click to see the all result, we can see here that we have got a lot of results. So what you have to do, you have to just export the table, input all these gene ontology terms onto the code, and the visualization will be done. I will make sure that this code is presented on my GitHub repository as well. But for now, for this paper, I'm just gonna leave it like this, because I think the biological processes are enough for this paper. So here we will leave this like this. So we have performed the distribution sample and expression density analysis, volcano plot visualization for the DEGs, domain analysis, protein interaction analysis, and now we have also performed the gene ontology and enrichment analysis. So let's look at the slides we got. So we have the enrichment categories, which is a biological process, cellular component, and molecular function. You can relate them and interact them to the lung cancer category. And then you can also create the Venn plot if you have a lot of genes. For example, I am just currently dealing with 2 or 3 of genes, but if you got a lot of genes, for example, you are coming down with the 20 up regulatory genes, 20 down regulatory genes, you have to create a Venn diagram that create the 3 enrichment analysis circular tab. So the tool that we use for this analysis is called Venni. Venni, you can search for this tool online, and then what you need is you need the number of IDs that were used in all the analysis, and then compare it, and the Venn diagram will be showed to you. So the tool name is Venni. You can look that up onto the database, and you can use that to create the Venn diagrams like this. Yes, here you can see the Venn is loaded. Now you can configure them according to your own need, and we have this, and if you can just see, we have the Venn diagram of each of the figures that we need. So you can change this into English layout as well if you want to, but I think this is enough for this lecture. So I just gonna leave it like there, so you can perform the gene ontology analysis like that. After that, we will move to the protein ligand interaction analysis and protein structure prediction. So that's it for this lecture. Thank you so much.